All right, let's look at what happens when we have a mutation. A gene mutation is any permanent change in the sequence of the bases in DNA. So sometimes these can have no effect at all on a protein activity at all. For example, remember this chart that we saw in the last chapter? It's showing base pairs. So 3 base pairs make a codon. And we're looking at those 3 base pairs and what amino acid they will lead to, because amino acids are those building blocks of proteins. So let's say we had TTG as our 3 base pairs for our codon. It would code for leucine according to this chart. If there were a gene mutation that resulted in TTA, so the G was turned to an A, that is a gene mutation, but it would have no effect at all on the protein activity because it would still code for the leucine amino acid, so no effect at all. However, if that G became a C, so instead of TTG, we would have a gene mutation to TTC, then that would change the amino acid to phenylalanine, and that would have an effect on that protein. Likely it would be a negative effect, but rarely you'll see some positive effects, and we'll talk about that when we talk about our evolution chapter. Most of the time it leads to complete inactivation of the protein, meaning it doesn't work at all. Sometimes it just alters the protein. And we'll look at the difference there as we go down. Germline mutations are mutations that occur in sex cells, and it's important to know the difference between germline mutations versus somatic mutations, which occur in body cells. Because germline mutations, since they're in sex cells, they affect the gametes, meaning they can be passed from parent to offspring. So these are mutations that will be passed down to children. However, somatic mutations, which occur in body cells, could be something like a mole. And that's not going to affect any offspring at all. Okay, let's look at some different mutations in everyday life. For example, red hair. Red hair color also comes along with freckles. It's due to mutations on both alleles of the MC1R gene, which have to do with melanin production or the coloring. So these, you have to have 2 recessive alleles in order to have red hair color. Shorter legs in these dogs, those are actually an altered copy. It's an extra altered copy of a growth factor gene. So these are both examples of mutations. Some mutations are spontaneous, like chemical changes in DNA that may lead to mispairing during the replication process, or the movement of entire pieces of DNA from one location to another on a chromosome. So this would be an entire piece just being swapped to a different place, and this may happen during either mitosis or meiosis actually, but remember in meiosis when we're talking about sex cells, you had entire pieces of DNA being swapped during the tetrads. And then you could also have replication errors. We kind of talked about this earlier when we talked about replication, where DNA polymerase, it does build new strands, it also proofreads new strands, and usually it corrects its errors, but sometimes it doesn't. Sometimes there's a mutation rate, and that rate is 1 for every 1 billion nucleotide pairs. So that's a lot, 1 in a billion. That rarely ever happens at all. However, if you think about it, how often replication happens, it happens more than you would think. And then there's induced mutations. Induced mutations are caused by mutagens such as radiation and organic chemicals. Many mutagens are also carcinogens or cancer-causing. Environmental mutagens can be UV radiation. This is sunlight. We all know about tobacco smoke, and the list goes on and on. There are many, many, many carcinogens. This is an example of how UV radiation is a mutagen. What UV radiation actually does is it causes something called a thymine-thymine dimer, meaning that, remember, A goes with T, G goes with C? Well, UV radiation can cause DNA to break, and it causes the thymine and the thymine on one strand to bind together. So it will break it, causing these thymines to form a bond. Okay. It's called a thymine-thymine dimer, and that's just from sunlight. Okay, so what are different types of mutations? There's two main types that we are going to focus on. The first one is a point mutation, and just like it sounds, it's where there's a change in just one point. Only one base has changed. It's a single DNA nucleotide change, a change in one codon to a different codon. Remember, codons are 3 pairs. The effects on the proteins will vary, just like I showed you on that chart before. Where was that? Right here. Okay. Right here where we talked about how you could have TTG moving to TTA and there'd be no effect. Those are examples of point mutations. You just have one change. Right here we have, it used to be CTT and it went to CAT, just 1 point change. Everything before and everything after that would be the same. And in this case, it did lead to a different amino acid. If we were going to put this into a sentence, we could say normal would be, we'd have to have them in, we read in groups of 3. So all of our words have to be 3 letters. So we have the cat ate the rat. That makes sense. And after the mutation, it would be the CAA ate the rat. And that's missense. Everything makes sense except for that one word that was changed. Everything else makes sense. Now compare that to a frame shift mutation. In a frame shift mutation, we have 1 or 2 nucleotides are either added or deleted. So entire sections added or deleted. The protein is always rendered non-functional if you have 1 or 2 nucleotides added. Because remember, our DNA is read in codons that are 3 base pairs long. So if we put it into the sentence again, we have the cat ate the rat. Now I am going to take out the C. We're going to remove 1 base pair, so deletion. So now it's the, that makes sense, but then after the mutation where we remove the C, we have atta tetrarat. Makes no sense at all. Nothing makes sense after that because we've disrupted our 3 base pairs. We've disrupted our codons. And this is going to be a nonsense, non-functional protein. We can also insert an extra C, and inserting the extra C still disrupts the codons, the pairs of 3. So we will have the, and nothing makes sense after that. That's the difference between a frame shift mutation, which is going to be an insertion or a deletion, or a point mutation. Now, something to point out, if 3 base pairs are added or subtracted, or any multiple of 3, then the only thing that will change is 1 amino acid because everything before and after will still be the same. For example, if I were to delete all 3 of these base pairs right here, CAT, all 3 of these bases, then it would read the ate the rat. So the only thing that would change would be this 1 amino acid. But normally a frame shift mutation would be non-functional. Let's look at some diseases that are caused by mutations. Right here we're looking at Tay-Sachs disease. Now, Tay-Sachs disease is a mutated enzyme caused by a frame shift mutation along a metabolic pathway that breaks down fatty substances in the cell membrane. So we're looking at a metabolic pathway, meaning you need an enzyme at each one of these steps. Enzymes are proteins, so each enzyme is going to have its own gene. And so if a mutation happens in any one of those genes, it'll cause a problem with any one of those enzymes. The whole entire metabolic pathway will shut down. So Tay-Sachs disease is where we have a problem with the enzymes right here. That is going to convert this GM2 ganglioside into this chemical right here. And so this cannot happen. This ganglioside is just going to build up. And the fatty acid substance is not going to break down. Meaning there's too much fatty acid in this cell, and it's going to affect the function of the nerve cells. It does lead to death because your nerve cells cannot communicate with each other. So that's one example right there. And there you can see it, metabolic pathways have many, many, many different steps in them. So there's many places where something can go wrong. There's actually a lot of research in this field looking at how metabolic pathways work and how different diseases are caused by problems within these metabolic pathways. Another example is a disorder called PKU. This is where phenylalanine actually builds up in the system, and it can cause an intellectual disability. But how phenylalanine builds up in the system is we have a problem with this enzyme right here. So this is only a small part of a metabolic pathway, and it leads to the production of melanin. And melanin is going to be the coloring within the skin and hair. So phenylalanine should be converted by this enzyme into tyrosine, which is then converted into this enzyme by melanin. But since this enzyme has a mutation, this phenylalanine just builds up in the system, and that actually is a toxin that will lead to intellectual disabilities. So there are many careers that you can follow that actually deal with the different metabolic pathways and diseases that are caused by mutations within those. All right, so that's what we're going to look at with mutations. Make sure that you know how mutations are caught. Let me show you right here. Different causes of mutations. So we have spontaneous and induced. And make sure that you know the difference between point mutations, frame shift mutations, and the different effects of gene mutation. So sometimes there's no effect at all. Sometimes it just alters the protein activity, and sometimes it completely inactivates the protein. All right, thank you guys. I'm going to put Amiibo Sisters up for this as well.